Okay, lecture 27. We're finishing up on reproduction, and I want you to turn to plate 160, regulation of sex determination, sexual development. Well, I think you're all right. We're diploid. Double X is a female, XY male. So the determination of sex really lies on the Y chromosome. And that Y chromosome has to come from the male. So when we talk about the determination of sex, it's a function of which sperm makes it into the egg. Is it the X or the Y? And in fact, X and Y sperm show functional differences. Now think about the potential for this. If you're able to differentiate between the X and Y chromosome, X and Y carrying sperm, then you could select for the gender of the offspring. Might lead to some problems, right? You know, the cultures that are very male-dominated only want males. They would then allow you to select those and from a human standpoint. But think about animal production. What group of animals is really heavily focused on females? The dairy industry. And in fact, when you see a female calf come out, that's much more money in your pocket, potentially, than a male. All a male is going to do is going to go for slaughter. There's some money to be had there, but by and large, it's the female that's going to make money. And so you might imagine that there's a fair amount of work in terms of trying to select X and Y bearing gametes. So you have male versus female. You have undifferentiated gonads. So if we go and we look on plate 160, we have the developing testes and the developing ovary. In fact, at birth, even if it's male, there is relatively little gonadal differentiation. Takes a number of weeks for the gonads to actually start developing. So consider a primordial gonad is composed of two layers, an outer cortex and an outer medulla, and that's represented right here. In a developing, if it's a male and the testes are going to develop, it is a development of the medulla. So the inner portion develops. If it is a developing female, it is the cortex which develops. So again, an undifferentiated gonad that looks the same. Well, what determines whether it goes to a male or a female? That has to lie in the Y chromosome. In fact, there is a gene, the sex-related region of the Y chromosome, a SRY gene. That gene codes for the transcription of a protein, SRY, which is important in modulating and stimulating the development of the gonad into the testes. So the Y chromosome induces testicular formation through this SRY. If there's no SRY gene, no SRY protein, then de facto you have development of a female gonad. So this process occurs in the absence of an SRY gene. So it's kind of like, well, we're all females, at least gonadally, unless we have the SRY gene. As the testes starts to develop, it is producing testosterone and a Mullerian inhibiting substance. Mullerian ducts are the progenitors to the fallopian tubes in the uterus, and so the Mullerian inhibiting substance suppresses the development of the female genitalia. The testosterone then goes on to stimulate the development of the testes, and at puberty, it'll lead to the secondary sexual characteristics of the male. Part of the action, cell, remember, if we refer back, what cells produce testosterone? Those are the Leydig cells of the testes. The Mullerian inhibitory substance is produced by the Sertoli cells. We talked about the Sertoli cells, right? They sit down within the matrix of in the seminiferous tubules. One of the important things they did in a mature male or in a post-pubescent male was secrete androgen-binding protein. That was very important in the further development and continual development of the spermatogonia. Well, now in the neonate, the Sertoli cells are producing this inhibitory substance which assures that the gonad develops into the testes. In addition, the testosterone acts on the hypothalamus. Critical. Hypothalamic functions in body temperature regulation, blood sugar regulation, sexual behavior, appetite, and now we see it plays a critical role in sexual development. Testosterone acts on the hypothalamus to ultimately produce releases of GnRH, gonadotropin releasing hormone, that are relatively continuous in fashion. That is, males are always producing some GnRH. So they've drawn these arrows, these very strong arrows, kind of indicating that testosterone stimulates the hypothalamus to release its gonadotropic hormone and GnRH. GnRH then goes and causes releases from the anterior pituitary of FSH and LH in a relatively continuous cycle. Well, look what happens then in the female is, in the absence of testosterone, there are surges in the GnRH and pulses then in the release of FSH and LH. And we talked about how important the pulsatile nature, the frequency and magnitude of those pulses were in setting the reproductive cycle. And that then is coming through the hypothalamus. Okay, plate 161 that I have on computer. So, excellent. So let's talk about fertility and what influences fertility. And I'm going to do this from the perspective of like a breeding soundness exam. So one of my responsibilities as veterinarian is to do breeding soundness exams in our bulls. We raise a number of bulls. I think this year we may have 30 that are going up for the bull sales. Well, so here's an example in which the male is very important in terms of the breeding process. We've taken all those unwanted males, we've castrated them, we're going to eat them later, but now the bulls are going to provide semen for the breeding stock. Is this animal capable of breeding? The bottom line, the success of mating is ultimately going to be determined in having a calf on the ground. Well, that's almost a year's time, over 300 days from mating to delivery. Wouldn't it be better? And that also has, the female plays a role in that. There may be a problem with the female reproductively in that she may have a perfectly viable fetus and then rejection somewhere along the line. Well, that is a breeding soundness problem with the female, not necessarily with the male. So how do we approach the breeding soundness of a male? We don't want to spend a year finding out whether this animal is capable of producing offspring. We want some insight on this animal right now. So things we start to look at that certainly affect efficiency and effectiveness in breeding. Do they have sperm or not? We talked about body temperature as being one of the major reasons for decreasing the number of viable sperm. So these animals are producing sperm, they just happen to be dead. They got killed by heat. Another, we had a stallion on campus who, as it got older, so we know something about age, was affecting this animal's GnRH. So this stallion was producing fewer number of sperm. So we see sperm number has an important influence on fertility. as an individual becomes infertile, or in a non-fertile individual, these numbers are very low or non-existent. There may be a change in the sperm morphology. So these kind of represent sperm that have lost the tail. Well, that's not going to move very effectively. Double head, no head. Well, here's a tail without any DNA. That's not going to work. Here's a sperm with double head. So a second thing that we do is when we collect an ejaculate from these animals, we recognize, first of all, do they have adequate numbers? Also, appropriate morphology. We look at sperm morphology. What are the number of abnormal sperm present? Something that isn't expressed in this is motility. So we also do a motility exam. That becomes important in distinguishing bad sperm that are bad because they've been killed off by heat. So you may have large numbers of sperm, but they will be amotile. A good breeding bull will have a large number of sperm, and there will be a high percentage that are motile, 90% are moving, and very little morphological problems. Sperm production is a rate-limiting process. You can only produce effective sperm at a certain rate. Sperm number drops off with the number of ejaculates. This becomes very important if you're trying to manage a breeding farm, and you recognize that sperm production is not unlimited. In stallions, that will have a profound effect on the number of bookings or the number of matings that you will have for a male. Factors affecting male fertility. Males tend to produce sperm pretty effectively throughout their career. We know that's not true with females, right? Spermatogonia develop throughout the life of the male. In the female, the number of oocytes is limited at birth, so you only have a certain number. So what we find with age is decreasing. Here's eggs per ovary, demonstrating that as you continue cycling throughout your lifespan, you reach a point which is referred to as menopause, in which there are no eggs or very few eggs being produced. Now, we can kind of modulate that. We can give these individuals estrogen therapy, and if you want the development of the follicle, you want to help maintain the viability of that ovary, you can give LH, FSH injections and HCG injections to artificially push them into that. That becomes important if you have a very expensive mare, very expensive cow, and you want her to continue producing eggs because they're valuable. You can start doing hormone replacement therapy. Contraception is a big issue, both in humans and in our domestic animals. In humans, they talk about the rhythm method, so not a big issue in our domestic animals. We have trouble in determining when these animals are in estrus. When are they receptive? This is the time that we can use to deal with a potential pregnancy. One of the issues of estrus in domestic animals is an aggression or responsiveness of the females to approaches by the males. Responses that might, in fact, damage the male. If you've seen them out teasing the mares, they'll bring a mare and a stallion in. If that mare is not sexually receptive, if she's not in estrus, she is interested in kicking the head off the stallion. Not good to be a stallion. When she is in estrus, when she's receptive, she will receive the male. Well, think about taking that horse, that mare, to a show. She is approaching the estrus cycle and is not yet receiving the mare or the stallion. She can be very aggressive. Conversely, if she goes into heat, she may attract the stallions, and the stallions then become very aggressive with one another, trying to make the mating. And if you happen to be in the way, it can be very dangerous. So we see a fair amount and effort at regulation, at contraception, particularly in show animals. What hormone is the hormone of pregnancy? Progesterone. So synthetic progestins are used characteristically to keep domestic animals from coming into heat. You don't want them to show estrus. You want them to think metabolically that they are in a pregnant state. And you recall that the hormone of pregnancy is progestin. So there are on the market now the synthetic progestins, oral and injectable for our domestic animals. We have particular concerns because a lot of our students are female. They're handling oil-based progestins. So if you reach for one of those progestins and you have an ungloved hand, you run the risk of taking up lipid, steroid hormone, can move, in fact, through the skin and be picked up. Well, if you're pregnant, big dose of progesterone may not be a problem, but it can also. What if you're trying to get pregnant and you're taking progestins? Can be a problem. The other common contraceptive is, in fact, sterilization. So we see that in domestic animals, particularly dogs and cats. The removal of unwanted pregnancies, removal of the gonads. In males, you castrate, you remove the testes. That removes viable sperm. It removes testosterone. The reduction in testosterone can do a good job at reducing libido and aggression. So behaviors associated with testosterone are decreased. And those are probably good things for many of our domestic animals. In the female, removal of the ovaries removes the source of oocytes. Generally what happens, though, is when they take the ovaries, they also take the uterus. Well, the removal of the ovaries is going to remove the oocytes. It's also going to remove the estrogens and the progestins. So these animals will no longer cycle. They will not go through estrus or heat. So some of those behaviors disappear. So why take the uterus? We talk about an ovario hysterectomy. So otomy, cutting out the hysterical portion or the uterus, and removing the ovaries. Well, in terms of sterilization, you do that by removing the ovaries. The point of taking the uterus is, at that point, the uterus has no function. It's extra tissue, if you will. You have extra tissue that in some future date may develop problems. You may get an inflamed uterus even in the absence of sexual stimulation and sexual reproduction. You may get tumors associated with that tissue. So the philosophy has been, well, I'm removing the ovaries, let me take that useless tissue and remove it from the body. We are seeing, though, that there is a trend to just removing the ovaries in very simple laparoscopic procedures. And we'll see how that progresses. The idea that you go in, what that does is that minimizes post-surgical trauma, a huge issue in veterinary medicine. So that concludes the reproduction portion of the lecture. So what I'd like to do is shift gears. We're going to review a little metabolic physiology. So I want you to turn to plate 130, if you would. We are not going to cover everything in the book. In particular, I have kind of short shrifted you on the neural circuitry. we'll leave that for another time. Some of this should be review. So let's start back then and talk about major substrates, carbohydrates. 3 examples of monosaccharides, or simple sugars. The one we have generally focused on is glucose. Galactose and fructose are other examples of monosaccharides. The transporters are for these, the transporters in the gut, right, are focused on primarily on these carbohydrates. We all know about sucrose. This is table sugar, a very common form. It is a disaccharide. The polysaccharides are linkages of these different mono and disaccharides to form long chains. Nutritionally or dietarily, we see these as starches. The long chain polysaccharide that is the storage form in the body is glycogen. Here is the stomach. Food coming into the stomach through the intestines. You have the starches that are being broken down by the amylases, the disaccharides being broken down, the monosaccharides being reabsorbed or being absorbed through the gut. And so what you have here is all of these little glucose moieties, these monosaccharides, that are being that are brought from the portal vein to the liver cell. The hepatic portal vein takes blood from the capillaries of the gut and delivers it to the capillaries of the liver. So this represents the exchange, the sinusoids and capillaries of the liver, where glucose is moved into the liver cell and converted to glycogen. So there are enzymes that you all learn in biochemistry that take the glucose and put it in the storage form of glycogen. This glycogen then can, during times of fasting, release the glucose for output into the blood to go to things like brain, muscle, and heart, providing the carbohydrate nutrients that are required. Neural tissue has an obligatory requirement for glucose. Red cells, they don't have any mitochondria. They got to use glucose. So we see a couple of tissues that have a very important dependence on delivery of normal glucose concentrations. Some of that is coming from glycogen. In the liver, some of the glucose is made from proteins and amino acids. So we're able to break down proteins then and produce glucose. Now, again, no storage forms for proteins. These proteins are structural. They're building components of the body. But in starvation, when you have no dietary intake of glucose, you need to be producing glucose. Glycogen stores are limited. So one of the ways you make new glucose during this starvation is you break down muscle tissue. You can get some glucose from glycerol. Fatty acids do not go to glucose, but remember that triglycerides are glycerol and 3 fatty acids. The free fatty acids are a great energy source. We'll talk about them in a bit. But the glycerol can be converted into glucose. So again, during fasting or starvation, you start breaking down fats in the liver, converting them to fatty acids. The fatty acids are going to go out into the circulation. The glycerol is going to be taken back and converted to glucose. Also, in the adipose tissue, the glycerol that's being produced by the breakdown of fats in the adipose tissue circulates through, and it can get picked up by the liver. So think of this liver as kind of poised. It's really there, importantly defending these glucose concentrations. So glucose goes out. Here, down here is our metabolizing muscle cell. Glucose is taken in. In the absence of oxygen, glucose is converted to lactic acid. We talked about lactic acid. If oxygen is present, we can funnel this glucose through the Krebs cycle and produce lots of ATP. In the event that we are oxygen limited, or we have a high demand for ATP production, lactate is produced. What happens to it? Some of it is converted in the muscle, and this is not demonstrated in this picture. Rather, what they show is the lactate going out of the muscle into the blood, back to the liver. This is what is referred to as the Cori cycle, the biochemist that identified it. This lactate then is converted back to glucose. So look at all of this energy and function of producing and maintaining glucose. Brain relies principally on glucose, although in fact it can use some ketones. But because of this high dependence on glucose, we make the neural tissue insensitive to insulin. We talked about insulin sensitivity. Insulin was required for the uptake of glucose into cells. Certainly works at the muscle level, but not at the neural tissue. At the neural tissue, you don't want glucose. You want glucose to be freely available for that tissue. All right, what about neural regulation of blood sugar? This should be some review as well. Regulation of blood sugar, what is called a glucostat, and that's just kind of a fancy word for maintaining a static or regulation of glucose. It's lodged in the hypothalamus. What are the hormones that influence glucose? Well, we've talked about a number of them, kind of demonstrated here. The hypothalamus has input down to the anterior pituitary. Hormones that were important in the regulation of glucose. Remember, growth hormone was a hyperglycemic compound. So is cortisol. Well, the release of cortisol is manifested by the release of ACTH. We talked about the important role of norepinephrine and epinephrine in the breakdown of glycogen. So here's the sympathetic nervous system causing the release of norepinephrine at the level of the adipose tissue, and epinephrine, which acts on the adipose tissue, and it also acts on muscle tissue. What else does the hypothalamus do? Satiety, eating. It drives hunger. And you said, like, I'm really, really hungry. Well, part of that is your brain sensing concentrations of glucose. And when you are hypoglycemic, that is perceived by the glucostat in the hypothalamus, and that then drives higher centers that says, I'm hungry. And the old eyeballs turn towards Carl's Jr. So part of that is not just hormonal control, but also neural control to stimulate food uptake. We talked about cortisol. What other hormone have we failed to talk about? Glucagon. All right, we don't see the pancreas in here. Glucagon is another hyperglycemic hormone. So in response to decreased levels of glucose, glucagon goes up. a lot of hormones then focusing on elevating glucose, only one hormone focusing on a decrease in glucose. And neural tissue is relatively insensitive to that. Plate 132 is an extension of that. Hormones that are related to the measurement or control of blood glucose. Again, hormones that we're all familiar with. We saw about the neural release of ACTH, its stimulation in cortisol production. Here's the pancreas with glucagon. Here's growth hormone repeated. Hormones that lower blood glucose level. We said there was really only one that was involved in decreasing blood glucose, and that was insulin. In your book, it talks about T4 and T3 as being hormones that lower blood glucose level. I think it's a bit confusing because how do they do that? What T3 and T4 do is they drive metabolic rate. Hyperthyroid individuals, high levels of T4 and T3, elevated metabolic rates. That consumes more glucose. So in fact, T4 and T3 are not acting to regulate glucose levels. They're acting to consume them through increase in metabolic rate. Metabolism of fat, plate 133. Storage form of fats are the triglycerides. Triglycerides are composed of 3 fatty acids, hence the term triglyceride, and glycerol. Glycerol is a carbohydrate moiety. Remember, we can make glucose from glycerol. And fats. Storage form, during starvation, triglycerides are broken down to give you fatty acids and glycerol. Components of fat metabolism. We talked about dietary fats. Remember, chylomicrons. Those are fats that are enveloped in protein coats, transported through the lymphatics, dumped out into the blood supply. So this shows this little chylomicrons being dumped out, moving towards the adipose tissue cell, fat cells. Lipases, membrane-bound lipases, break down the chylomicrons, shuttle the glycerol and the fats in, and then you reform the triglycerides. So lipid adipose cells are nothing but sacs of triglycerides. They sit there, very high energy density, waiting for metabolic processes to demand energy, working muscle. Fats take small space, liberate a lot of energy. There's no water requirement for the storage of fats, or very little, so you don't have to hydrate the molecule. And fats have higher energy densities. So if you take a gram of carbohydrate, dry, no water associated with it, and a gram of lipid, dry, again, no water associated with it, and you measure the energy content, the potential for ATP production, it's twice the amount in adipose tissue. The breakdown of triglycerides to fatty acids, and fatty acids then to metabolizable substances like ketones is a process called beta oxidation. I don't want this to be a biochemistry course, but I do want you to be familiar with some of these terms, which in fact to me make biochemistry infinitely more digestible, right, to kind of see how these enzymes and how these compounds are working. If we look here in this cell, we see the glycogen being broken down, glycerol. Storage form of fats, breakdown of fats, and production of ketone bodies. A consequence of fat metabolism is the production of ketone bodies. In severely, remember in diabetics we talked about the fact that there was so much fat metabolism and breakdown of fats that you were producing excess amounts of ketones. Ketones that appeared in the urine, a ketouria, and ketones that appeared in the blood, a ketonemia. Well, too high concentrations, that's toxic. But we tend to use ketone bodies all the time. They are probably most pronounced in diabetics and also in starvation. So these become important energy substrates to a starving individual. Fat utilization by the muscle, yes, it does. We've talked about fats in the muscle, so this is the heart and muscle cell down here. Ketone bodies, fats, both can come into this cell. Beta oxidation also occurring in the muscle cell, the breakdown and feeding into the Krebs cycle to produce lots of ATP. So our muscles are very effective at utilizing fats. We can change, by changing fiber types, by exercise, we can focus and change fiber types' capability for metabolizing fat. Endurance runners, great way to train up your muscle cell to metabolize fats. (134) regulation of fat metabolism. Doesn't look yet. Hypothalamus, again involved in the regulation of fat metabolism. Regulation of glucose, regulation of fat metabolism. Hormones and neural control of that, very similar to glucose. Sympathetic nervous system. Remember, the sympathetic nervous system was activated to break down glycogen to glucose. You needed to provide the energy for a flight or fright situation through norepinephrine, through epinephrine. Similarly, fat metabolism. Lipases are activated by epinephrine and norepinephrine. Growth hormone, important in providing glucose for a growing body. Growth hormone, important in providing lipids for growth. Cortisol, stress hormone. What do you need when you're stressed? I need glucose. What else do you need? You need fats for energy. So we see parallel. All these same hormones that are acting on cells to make glucose available for working muscle, for the heart, and for the brain. Similarly, energy from fats. One hormone that I do want you to be familiar with is leptin. Leptin is received a huge amount of interest because it is a hormone which is involved in inhibiting the hypothalamus. Is a compound that will induce satiety or make you satiated. So you can imagine the interest in this hormone, like, I need to take a special diet pill. I don't want to be hungry. What can I do to reduce my cravings? We talked about this, the central nervous system. It's being driven in part by this hypothalamus. What if you satiated your hypothalamus? Say, look, I've got plenty of energy. I don't need to eat anymore. I can reduce my fat intake. Leptin has been a compound of interest. We haven't seen it reach GNC yet, but our understanding of hormonal and nervous control of metabolism, really a lot of money, a lot of interest to be made. I think what I'd like to do is stop there, and then we'll pick up plate 135.